In this class, we are going to start molecular genetics, that is the molecular basis of inheritance or heredity. So in the previous chapter, we have studied about various genetic processes, and in this chapter, we are going to study what are those processes and how those are occurring at a molecular level or genetic level. So the main things about the previous chapter and this chapter is very much related, but the difference is that we studied those phenomena on a macro level in the previous chapter, and in this chapter, we are going to study those things on a molecular level. So we are going to start this chapter. The first thing about this chapter is that we know that our main genetic material is DNA in almost every living thing. Apart from some riboviruses, every living thing has the main genetic material as DNA. So we now know this as a fact, but lots of years ago, or maybe a century ago, people were not convinced or confirmed about that DNA is the genetic material. So there was a lot of research and discoveries going on around these genetic phenomena and genetic mechanisms which lead to transmission of hereditary characters. So lots of scientists were working on them, but none of them could actually verify what is the actual nature of the genetic material. So we will first have to understand how and why people or the scientists concluded that DNA was the genetic material, and it was the main responsible molecule for transmission of genetic characters. So we'll first study that. So there were many scientists who were researching on this phenomena and searching for the genetic material, but we mainly have to study about the experiments of three. Out of them, the first experiment that we have to study in this topic is the Frederick Griffith's experiment. So he was experimenting on a lot of things, but one of that was Streptococcus pneumoniae, or that is also called Diplococcus. So this transformation is a process that occurs in bacteria by which it changes its genetic constitution. So Griffith used this phenomena to clarify, or he could not actually conclude, but he did get an indication about the biochemical nature of DNA or the genetic material. So we'll study his experiments, but before that we'll have to study what is transformation. So what happens here is that bacteria generally has the ability to absorb DNA or genes of its dead relatives. That is, we'll study in this section about a bacteria called Diplococcus or Streptococcus pneumoniae. So that bacteria has the ability to absorb DNA or genes from its dead relatives, because the bacteria has many strains like smooth and rough strains. So we'll study about those. So those rough or smooth strains do have the ability to absorb genes or take genes or DNA from the dead members of their own species or their own strain. So this is what happened in this case of Frederick Griffith's experiment. So this bacterial species has two strains, that is rough and smooth. So this rough was one non-virulent, or it did not cause any disease, but the smooth one was virulent, that it causes diseases. It was because this smooth secretes a mucilaginous sheath around itself, and therefore it is able to resist against the enemy's antibodies or WBCs, and this causes diseases where this does not cause any disease. So this rough is non-virulent. It produced rough colonies in culture. This smooth is virulent, that it is causes disease. Virulent means it causes disease, non-virulent means it cannot cause disease. It produces smooth colonies. So it is smooth because it secretes a certain mucilaginous sheath. This mucilaginous sheath is the one that protects it from the enemy's antibodies, and also this is the cause of this virulence nature of this smooth strain. So this smooth also had a lot of types, and this rough also had a lot of types. So this Griffith experimented on the R2 strain, that is rough 2 strand, and S3 strand, that is the smooth 3 strand, and he took those two bacteria and injected into mice and saw the results. So this smooth one can cause a deadly disease that is called pneumonia. That is why its name is also pneumonia. So what happens is that this smooth, when it was injected into mice, and when it was alive, it caused disease in mice and caused death of the mice. But when this rough strain was used, that is the R2 strain was used, then it could not cause the disease to the mice, and therefore mice lived. So these two had different effect on the mice. So it were as expected, because it is virulent, it can cause disease, mice will die. It cannot cause disease, mice will not die. So these two results were very expected. But what happened when these two were combined, that is what caused the Griffith's experiment to conclude something about the genetic material. Now we'll study his experiments. So when Griffith injected R2 into mice, then mice lived. So it was expected R2 is not a virulent, so mice would have lived. But when he injected S3, then mice died. But when he injected S3, which was killed before, that is when Griffith injected dead S3 Diplococcus bacteria into the mice, then mice did not die, because when S3 is already dead, how can it cause any disease? So therefore, when S3 was injected heat killed, in the heat killed form, mice lived. In this case, mice lived. So this was also expected, because a dead bacteria cannot cause any disease. So these three results were very expected, and there was nothing a bit off the way about these three results. What was the fourth experiment, and that is what caused those conclusions. So this is what is the main point in this whole discussion. So when S3 is heat killed, it does not cause any disease. But when R2 is alive, it also does not cause any disease. Therefore, mice lived in both these cases. But when these two are used in combination, so that is causing some mice to die, whereas some mice did not die. So from here, we can conclude that there was some change in either S3 or R2 that caused some mice to die, because R2 alone would not kill any mice, because we have already seen in the first experiment. So we have to study what and how that could have occurred. So we have to know that this S3 was heat killed. So it was even though it was killed, but it did contain its own genetic material. That is, the heat killed S3 was having its own DNA, its own RNA, or whatever be the biomolecules. Although it was dead, but it was not devoid of its molecules. And this R2 alone will never cause a disease. That's a fact. But when this R2 was with S3, some R2 did become like S3. That is, whatever be the molecules of S3 in this case that caused the mice to die, that molecule was synthesized by R2 in the presence of heat killed S3. So this is the main point over here, that this died mice did die because of some biochemicals secreted by S3, and R2 does not secrete those three, and S3, since it is already killed, so a killed bacteria will not secrete any substance on its own. So who would have secreted that biochemical? So obviously then R2 would have secreted that biochemical. And from where did it secrete? Because it does not have any property for that. So it absorbed some of its genes or its DNA and became like S3. So this is the main conclusion that we can draw from this. That is, S3 genes of this bacteria were absorbed by R2. So R2 only became pathogenic after it was mixed with S3 dead bacteria, not with the when it was alone. So this is the conclusion. So we are not writing here DNA because Griffith was not certain that it was DNA which caused the transformation. So he concluded that there was some biochemical that of S3 that were absorbed by R2 and which enabled R2 to become pathogenic, that is R2 became like S3 and caused the disease, therefore some mice died. And the second thing is some mice lived. So we have to understand that not all of the R2 bacteria were transformed into the S3 bacteria, therefore the bacteria of R2 which were not transformed, because of those bacteria this some mice lived, and only those R2 who were transformed into S3, those caused the death of some of the mice. So this is the main conclusion. So therefore not all of the R2 bacteria were transformed into the S3 bacteria. So this is the simple version of the Griffith's experiment. So this was the first experiment that gave scientists some hint about the biochemical nature, but this did not provide any concrete proof that which chemical caused this transformation. So we have to study more experiments about this also. So the second experiment that we have to study was of three scientists, that is Avery, McLeod and McCarthy. So these three scientists rediscovered, these three scientists caused a lot of variations in the Griffith's experiment, and these scientists hence concluded that it was DNA that was causing the transformation. So these three scientists experiment, that is Avery, McLeod and McCarthy experiments, were not very different from Griffith. They just took this experiment a step forward. So these scientists isolated the chemicals from the bacteria of R2 and S3. So they isolated the chemicals of S3 which was dead, and they divided those chemicals into three types. So these people used the S3 dead bacteria and extracted from them proteins, RNA and DNA. So from this bacteria these things were extracted, and then they again conducted the experiment done by Griffith. So we have to study what they did. So they took R2 bacteria and they put proteins of here of S3. Here no transformation occurred, that is the R2 bacteria could not kill the mice. So we are not discussing about which mice was killed and which mice was not killed. So we are just concerned ki how many R2 were or whether in which case R2 was transformed into S3 or not. So we first took the proteins of S3 and then mixed it with R2, but still it does not cause any transformation. Then R2 was taken with the RNA of S3. But here also no transformation occurred, that is R2 did not change into S3 and cause disease. Now in the third case, R2 was taken and DNA of S3 plus DNase enzyme also was taken. So here also no transformation occurred. So what we have to study here is that this DNase enzyme can digest the DNA, RNase enzyme can digest RNA, protease enzyme can digest the proteins. So whether protease enzyme were used in this case or not, no transformation was observed. Whereas here whether RNAs were used or not, in no case whether RNA or RNA with RNase was used, no transformation occurred here. But here when DNA was used with DNase, here it did not cause any transformation. In the fourth case, only the DNA of S3 was used. R2 plus DNA of S3. So in this case, transformation occurred. So we now can conclude that it was the DNA which caused transformation of R2 bacteria into S3 in Griffith's experiment. So these people were the real pioneers who for the first time they concluded that DNA was the genetic material, because they provided a concrete proof about this from their experiment. So this was the first case, this was the second, this was the third, and this was the fourth. So this transformation which occurred here provided the main source of this information that DNA is the genetic material. Apart from that, we also have to study the experiments of Hershey and Chase. So they provided certain evidences which were undisputable, and everybody had to accept that the DNA was the genetic material. So So these people used E. coli and T2 bacteriophage to understand what cause transformation, that is how DNA is transferred from the virus to the bacteria, or what is the real nature of biochemical or the genetic material. So we have to study their experiments. So this virus infects the bacterial cell, and thereafter it produces its own virus particles inside the E. coli, and that is called lytic cycle. But here we are not concerned with that lytic cycle. Here we are concerned what is being transferred from the virus to the bacteria. So in the beginning of their experiment, we have to study what these people did. So these two chemicals were used first in the case of E. coli. So when E. coli is grown on a substance that contains phosphorus 35, it's a radioactive isotope, and sulfur 32, it is also a radioactive isotope. So when these two substances are fed to the bacteria, then we know from our biomolecules chapter that DNA contains phosphorus, that is a phosphate group is contained in every nucleotide of DNA. So this phosphorus 35 will be inducted in the genetic material of bacteria. And from here we also know that phosphorus is generally not contained inside proteins. There are exceptions where phosphorus is included in proteins, that is after some post-translational modifications. But we are not concerned here with that. Apart from this radioactive sulfur, we know from our biomolecules chapter that amino acids cysteine and methionine contain sulfur. So if we use this sulfur, then that sulfur would be inducted in the proteins of E. coli. So this radioactive was in the DNA of E. coli, and this is in the protein of that is E. coli. And now this bacteria was allowed to be infected with viruses. So in NCERT, it is given that virus were grown on a medium. So we first have to understand that virus is not a living thing. Virus is intermediate between a living and a non-living thing. So viruses are obligate parasites. That is, viruses cannot be grown on a culture medium, because viruses can only grow inside their host, that is the organisms that they infect. So according to that line of NCERT, it cannot be very sure that viruses could be easily grown on a plate. So the actual thing that happened is that the viruses were first infected in these bacteria, and these bacteria were first radio labeled, not what is given in NCERT that viruses were grown on a medium. Viruses cannot be grown on a culture plate or on a medium apart from a living organism which it infects. That is why viruses are called obligate acellular parasite. So these viruses, when they infected the radio labeled bacteria, then only they came to have phosphorus 35 in their DNA and sulfur 32. Both of these are radioactive in their protein code. So now these viruses were allowed to infect unradio labeled bacteria, that is normal bacteria. So this was the main beginning of the experiment. Now we will study what happened and what were the results of that experiment. So when first of all E. coli were radio labeled, then normal viruses infected that, and after that normal viruses infected those bacteria, the viruses themselves became radioactive, and then those radioactive viruses were allowed to infect the normal E. coli cells. That is, this E. coli was also divided into two groups, and one group was infected with this S2 containing virus, and the other group was infected with their P35 containing isotope. So this is the beginning of the experiment. This is not the main experiment. So the experiment starts from here. That is, one normal group of E. coli is infected with a radio labeled P35 containing virus, and now the other group of E. coli is infected with a virus containing S32 in its protein coat. So we'll now study how those viruses infected and what was their results. So now we will study about how those viruses infected the bacteria and what was their results. We can consider it a normal E. coli cell, and we will also take a normal other group of E. coli cell, and we will see what happens in each of those cases. So it's a normal E. coli cell, it's also a normal E. coli cell. And then it was infected with viruses, the bacteriophage, that is containing those radio labeled substances, and here also it was infected So in both of these cases, their DNA were different. That is, this DNA in this case was DNA with P35, that is radioactive DNA, and in this case the protein coat contained S32. Protein coat contained S32. Now this virus was infected this E. coli and this E. coli as well. So after their infection, those were separated by centrifugation and then they were analyzed. So after this, what happens is that we know that when virus infects a E. coli cell, its DNA enters the cell, whereas the outer protein coat remains exterior. So we'll have to study about those things in both these cases. So what happens here is that the viral DNA enters the bacterial DNA. So from here this viral DNA has entered into the bacterial DNA. Here also the DNA, it was normal DNA, that is it was not containing phosphorus 35, and here also what happens will that the same thing, that is the viral DNA will enter the E. coli cell. Here also protein coat will remain, here also the protein coat will remain. Now we are gonna separate these bacteria and viruses through centrifugation. So what happened that after centrifugation both the bacteria and viral protein coat was separated. It was observed that in this case तो यहाँ पर जो बैक्टीरिया सेपरेट हुए हैं वायरस से, तो ये जो बैक्टीरिया है रेड कलर का, ये बैक्टीरिया जो है वो रेडियो एक्टिव मिलता है. एंड व्हाट हैपेंस इन दिस केस इज दैट वायरस का डीएनए यहाँ पर आया है, दैट इज वायरल डीएनए हैज एंटर्ड इनटू द बैक्टीरिया, बट स्टिल the separated bacteria does not contain radioactive material, that is the protein coat contains a radioactive. So this was the final proof that DNA is the main genetic material in organisms. So what happens is in this case is that virus infects E. coli, its DNA enters into E. coli, its protein coat does not enter. So in this case its protein coat was S32 containing, therefore that was radioactive. Here viral protein coat was radioactive, that means that protein coat did not enter the E. coli. Here it was the bacteria which was radioactive, and what has entered from the virus into the bacteria? So it was viral DNA. So therefore this provided the final proof that DNA is the main genetic material, and DNA is what is transferred in this case, or in the transformation principle, or in normal reproductive processes. It is the DNA that carries the genetic information. So this was the normal and the final Hershey and Martha Chase experiment, and this was the final proof. After this we have now established that DNA is the main genetic material. After this we have to study about the properties of DNA. So the DNA is just like any polymer. Likewise, protein is a polymer of amino acids, glycogen is a polymer of glucose, cellulose is also a polymer, starch is also a polymer. So DNA is also a polymer. Here the monomeric units are called nucleotides, and we first have to study what are those nucleotides. So nucleotide contains three components. So it is contained of three substances. First one is a phosphate group. The other substance that it contains is a ribose sugar, or rather deoxyribose sugar. So this deoxy is necessary because we are studying DNA. The full form of DNA is deoxyribonucleic acid. So why it is called deoxy? Because in this ribose sugar we will later study a structure that the OH group at the second carbon is lacking, and that is why it is called deoxy. And the third thing that it contains is nitrogenous base. So nitrogenous bases are also of two types. The first one is called purine and the other is pyrimidine. In purine we have two types of bases. One is called guanine and the other is adenine. In pyrimidine we have two types of bases. First one is called cytosine and the other is called thymine. In RNA we will also study that in place of thymine there is something called uracil. So that is not contained here, but that is a part of RNA. So that is why I am writing in bracket here RNA. So this uracil is not in RNA, in DNA, it will only be present in RNA, and this thymine will be present in DNA. So these are the normal constituents of nucleotide, and those nucleotides are polymerized to form DNA, and DNA is a double-stranded polymer. We will later study how and why that double-stranded polymer is formed. So we first have to study the structure of these substances and how these are located or attached to each other. First of all, we will study about the ribose sugar. So this is deoxyribose sugar. This is present in DNA. That is, this is the main sugar part. These are the nitrogenous bases. The structure of these nitrogenous bases are really complex, and there is no direct questions from the structure of these nitrogenous bases in NEET. So we don't have to study the main structure of this part. We just have to know that these are nitrogenous containing bases. And this is a normal phosphate group that is from it is derived from orthophosphoric acid, that is H3PO4. So these are the main constituents of normal nucleotides. So we have to now study about how these things are attached. So we'll study that now. So since we have studied the structure, now we will see their attachment. We will study again the same thing. So I am denoting nitrogenous base by NB. So it is attached to the first carbon of ribose sugar. And here we have the hydrogen groups group over here. Here we have this H group, and also we have a CH2 group which is attached to a phosphate group. This is not phosphorus, this is a phosphate group, that is why it is being encircled. So this is the normal structure of a nucleotide. Nitrogenous base from here, it could be either of these bases, then a deoxyribose sugar, and then a phosphate group. So this is the normal structure of a single nucleotide. So this is a normal nucleotide, that is, it contains phosphate group, ribose sugar, and also a nitrogenous base. Now we have to study how this is polymerized, that is how one nucleotide it attached to a different nucleotide. So above and below it is attached to the similar nucleotide. So this is how the normal nucleotides are attached to each other. So we have to understand here that this phosphate group is forming a linkage with here also and a linkage with here also. So therefore this type of linkage is called phosphodiester bond. And we'll study now where are the numberings of the carbon atom. So this is the first carbon atom, this is the second carbon atom, this is the third one, fourth and fifth. So we have to study here that phosphate group is attached to fifth carbon at one end, and at the other nucleotide it is attached with the third carbon. And in this case we have the same thing. So this phosphate group is attached with the third carbon over here and also the fifth carbon over here. So this type of linkage is called phosphodiester linkage. And this nitrogenous base forms a bond with this carbon atom, and this bond is called as N-glycosidic linkage. So these are the two types of bond, and this bond is a normal bond. And these two bonds together constitute the normal nucleotide. So we have to study about those linkages. This linkage is called N-glycosidic linkage. This bond, that is one above and one below, this bond is called phosphodiester linkage. We can call it phosphodiester linkage, or we can also call it phosphodiester bond. So after this we have to look at one thing. This phosphate group at one end of the sugar phosphate chain is called the 5' end, because it is attached to the fifth carbon. This end is called 3' or because it is called it is attached to it has a normal, if there is no other ribose group after this, then it will have a normal OH group, but if there is another nucleotide attached, then it will have the normal phosphate group. So here there are no other nucleotides, so therefore we will draw here OH group. So this end is called 5' end, and this end, because this is the third carbon, it will be called 3' end. We have to study these polarity, that is from where to where these strands occur. So the normal DNA strand is studied in a 5' to 3' fashion. That is the normal order of the nucleotide chain is from 5' to 3'. So this is the normal direction. How these are defined? These are defined by the phosphate group and the OH group. Since there are no other nucleotides above it, so phosphorus is attached with the 5th carbon over here, and in this case this OH group is at the 3' or the 3rd carbon of this deoxyribose sugar. So what happens is that this OH group or this end is called the 3' end, and this is what is very important in the case of DNA. So this is the normal structure of nucleotide and the DNA chain. Now this could be extended to any length above and below. Apart from that, another DNA chain will have another similar structure, but this will be anti-parallel to this. Like here we have 5' to 3'. The other DNA strand here will have the 5' to 3' here. So therefore this is the direction of one chain. So therefore the other chain direction will be from this side to this side. So we have seen the molecular structure of a nucleotide. We now have to see about nucleosides and what are these composed and how these are made. So first of all, we'll have the nitrogenous bases. That is, the first one is adenine. When adenine combines with pentose sugar, that is ribose sugar or deoxyribose, deoxy is denoted by D. So D-ribose sugar, it is called adenosine. So when a nitrogenous base attaches with a ribose sugar, that is called as nucleoside. So this is the difference between a nucleotide and nucleoside. So this is a normal nitrogenous base. This is pentose sugar. And this is called nucleoside. When this nucleoside combines with a phosphate group, it forms something called nucleotide. So this is how these structures are made and how these are named. So when this adenosine combines with a phosphate group, it will form monophosphate. When this is combining with phosphate, it will form deoxyadenosine monophosphate. So it will be denoted in a short form like DAMP, D-adenosine monophosphate. So this is how these are attached. Adenine, when it attaches with ribose sugar, adenosine, and then when it phosphate groups combines, then adenosine monophosphate. Likewise, we can do it for the other nitrogenous bases, adenine, guanine, plus this sugar, it will form guanosine, and when it combines with a phosphate group, it will form D-guanosine monophosphate, that is DGMP. Likewise, we can do it for the other bases, cytosine, the same thing. Now it will be form cytidine. not cytosine. That is, it will form, now its nucleoside is called cytidine, and plus phosphate group, it will form D cytidine monophosphate, and it will be bracketed as DCMP. And the fourth one is thymine, then it will form thymidine, not thymosine. So these are the normal 4 types of nucleotide, that is DMP, DGMP, DCMP, DTMP. So these are the 4 nucleotides that are present in a DNA. So these 4 nucleotides undergo polymerization and ultimately form the main strand of DNA. Now apart from this, there is also called uracil, which will be present only in the case of RNA. So RNA uracil, since it is present in RNA, it does not contains deoxyribose sugar. It contains normal ribose sugar. So it forms a nucleoside with uridine, and it is named uridine, and here it will be called, it will be not called D, and it will be called uridine monophosphate, or directly uridine monophosphate, UMP. D will not be used over here because it is in the case of RNA, only in RNA, not in DNA. So these are the normal 5 types of nucleotide present in DNA or RNA, and these will form the basis. Apart from that, we will now study the normal chains and properties of DNA strands. So the 2 strands in a DNA chain is both are 2 helically coiled around each other. And what is a helix and what are the properties of that helix, we have to study now. So we first have an imaginary axis. The 2 DNA strands are not coiled along each other. They are literally coiled around a common axis, like this. So this is what happens in the case of DNA. So it gives rise to certain types of grooves and other part of those also. So we have to study about those parts and those strands. So this is a bit too close to each other. I'll draw another a bit wider diagram. To understand the parts, we have to draw a bit wider diagram. This is a better one. So these are 2 strands of DNA, and these 2 will be anti-parallel. That is, these will be complementary to each other. And we'll also have to study the various properties over here. So from where one DNA strand starts to where it completes one circle, so that is called as pitch. So from here to here it is called pitch. From here DNA completes one rotation around the axis and the length of this pitch is 34 angstrom or 3.4 nanometer. In one pitch there are 10 base pairs, that is 10 DNA strands, 10 nucleotides will be with complementarily to each other. So in between from here to here there are 10 base pairs, that is 10 nucleotides which are pairing with the other strand. So this is the normal structure. Here we have to understand that the distance between a normal nucleotide and a successive nucleotide is around 3.4 angstrom. So this distance is 3.4 angstrom. Apart from this, we also have to study a few things over here. We see that two types of grooves are created. This is called a minor groove and this is called a major groove. We have to label those also. So this area that we are seeing with green color, this is a groove and this is also a groove. So this one is called a minor groove. And this is a major groove. Apart from that, we also have to understand the thickness of this strand. That is, the whole strand is approximately 20 angstroms in thickness. This is around 20 angstroms. Here we also have to study the dimensions of this minor groove and this major groove. So this major groove is approximately 22 angstroms. And this minor groove is approximately 12 angstrom. These are the normal dimensions of DNA. So we will have to study about these properties, that is, this DNA strands are not coiled upon each other but they are stacked in a way that is a staircase like structure around a common axis. And this is the normal structure of a DNA helix. It contains 10 base pairs in one pitch, that is from here to here the length of the pitch is 34 angstrom, distance between two successive base pairs is 3.4 angstrom. So this is the normal property of DNA helix. We'll now study we'll have to study each of these properties in detail. We can also say it as properties of normal DNA, or we can also, since DNA is in the form of helix, we can also say it as property of DNA helix. So we have to understand that DNA is the largest molecule in the cell. So what the second statement means is that the two strands that we just drew, so that strand, that is if one strand is from 5' to 3' direction, the other complementary strand will be from 3' to 5' direction. So this is what is meant by anti-parallel. So those two DNA strands are not exactly coiled upon each other like this. We have just seen that these were coiled around the imaginary axis. That is, these were like this, not like this. This is what is meant by this statement. So the two strands of DNA are coiled around an imaginary axis. That is the axis we drew just before the slide. That axis was the imaginary axis of DNA. So we just had seen that the strands or the backbone of the DNA is formed of sugar phosphate group. That is the main strand part which we just draw, composed of sugar and phosphate, and in between those two strands these nitrogenous bases were projecting inward. That is they were almost perpendicular to the axis of the normal sugar phosphate backbone. So this is how those things are located in the DNA helix. So what happens here is that nitrogenous bases of one strand will form complementary base pairing with the nitrogenous bases of the other strand. So here what happens is that adenine forms a base pair with thymine. So this is how the base pairing takes place. Thymine always forms pairing with adenine, and C always, that is cytosine, forms with guanine. So these dotted lines, these are called hydrogen bonds. So here there are two hydrogen bonds, and here there are three hydrogen bonds. So this is what is happening over here. since here only 2 hydrogen bonds are present, here 3 hydrogen bonds are present, so it is but natural that it is difficult to break C from G. That is, the cytosine and guanine hydrogen bond will be stronger and it will melt at a higher temperature, and it will separate at a lower temperature. So this is the normal, these are the normal properties of the DNA strand. Apart from that, there is something called stacking of the planes of the base pair. So because of these stacking of the planes of the base pair, DNA is extra stable. That is, it confers stability to the normal double helix of DNA. So that property is also that we are not going into that detail in this crash course class. So these are the normal properties of DNA helix. Now we have to study about Chargaff's rule or law of base equivalence. So this is what is given in our NCERT. So what happens over this case is that a purine will always complement with a pyrimidine. That is, the molar ratio of both will be same. So the ratio of, since purine and pyrimidine is 1, that means the purine is equal to pyrimidine in terms of molar concentration or in terms of exact number. So therefore we also know that there are 2 types of purine, that is Ga, as well as there are 2 types of pyrimidine, that is cytosine and thymine. So their ratio will also be 1. So that means that if the molar ratio of G and A are combined, then that will be equal to C and T, because we later now we have already seen that C forms a bond with G. Here we also have seen that is G by C is equal to 1 and A by T is equal to 1. Because every adenine will form a complementary pairing with thymine and every guanine will form a complementary pairing with cytosine. So this is what happens over here. Now one thing is to be considered here, that if Ga and plus Ct, that is their ratio are 1, so could it be that A by T, A plus T or G plus C would be equal to 1? So that will never be the case. It is not equal to 1, but whatever be its value is fixed for a species. So this value of the A plus T plus G plus C is fixed for a species and it cannot change. Every species has this ratio in a fixed at a fixed value that cannot change within a species. So this is the normal Chargaff's rule. Now we have to study about the packaging of DNA in a cell. How that long DNA is packaged inside a cell. So these are the normal examples of certain organisms and the number of base pairs in them. So the length of the DNA is normally measured by the distance between the base pair and multiplying that with the normal number of base pairs. So we have to understand how that is calculated. So here, if we talk about length of human DNA, so that will be this number multiplied by 3.4 nanometer. So length of the human DNA, for example, so to find it we have to multiply this number by the base pair distance. 6 into 10 to the power 9. We have seen in the structure of DNA helix that between two successive base pair the distance is 3.4 angstrom. So if we convert angstrom into meter, we have 10 to the power minus 9 and these are the number of base pairs. So if we get the calculate, then the value will come around 2.2 meters. So you can imagine that our one cell, just one cell of human body contains DNA of 2.2 meters. So how is such a long DNA packed into a small nucleus of just 10 to the power 5 minus 5 meter? So this is the size of DNA and this is the dimension of a nucleus, that is your normal typical human nucleus. So how can such a small nucleus accommodate such a large size DNA? So this is what we have to study in the case of packaging of DNA. How this long DNA will be accommodated into this small DNA. So this occurs through a process and that is called packaging. Because of that only we can accommodate our DNA. So this is called packaging, and we have to study this packaging here. So we first have to study about packaging in prokaryotes. So it is very simple that in the prokaryote DNA is not having some proteins that is called histone proteins, but still it does have some proteins called polyamines. So in the case of humans, that proteins is called histone. So here histones are absent, but in place of histone there are certain proteins called polyamines, and it is these polyamines which are helping in the packaging of DNA in bacteria, and that composite structure is known as nucleoid or polychromosome, prochromosome. So in our NCERT we have this name, that is nucleoid. Apart from that, the other name for this is called a prochromosome. So this compaction occurs with the help of RNA as well as with the help of polyamines. So in a bacterial chromosome, polyamines will be there, RNA will also be there, and apart from that, those structures will help in holding of DNA. But now we have to study about packaging of DNA in eukaryotes in detail. So this is what we have to study in detail, and in NCERT it is also very well given. So here we have to study about certain proteins. So in the case of bacteria, we had a polyamines, but here we have histones. So these histones are basic proteins. Why these are basic? Because these are rich in basic amino acids, that is lysine and arginine. So what happens here is that these amino acids have a lot of positive charges in their side chains. So in 11th chapter biomolecules, we have studied the structure of amino acids, and there we had studied about the side chain as well as the carboxyl group as well as the OH group. So the side chain of these two amino acids contains positive charges, and that is what helps in the attraction of DNA, because DNA is a negatively charged molecule. We'll see that later. But here we have to study how those proteins are arranged. So these nucleosomes are formed by these histone octamers. So we have to study what are these histone octamers. So this nucleosome contains 5 types of histone proteins. So these histone molecules have 5 types over here. First one is H1, the other is H2A, then we have H2B, then we have H3, and then we have H4. So this H1 is not a part of these core. This is a part of something called linker DNA. We will study later what is linker DNA. Part of linker DNA. This H2A and this H2B are 2, and each of these are twice, that is 2 molecules of each are present. So this forms histone octamer or the new body. So the core of nucleosome is formed of these 8 histones, that is 2H2A, 2H2B, 2H3 and 2H4. So these form the histone octamer. So this is a spherical ball-like structure, this histone octamer, and around that the DNA strands wrap themselves, that is like this. If this is the new body or the histone octamer core, then DNA strand will wrap around itself like this. And this H1 protein forms the plugging part, that is, it will help in the plugging or attaching of this. So this red color structure that we see here is the H1 part. And this forms the histone octamer, or it is also called a new body, or we can call it histone octamer. And this is the DNA strand. So in this new body or the nucleosome, what happens is that approximately 1.75 turns are of DNA occur. That is, DNA comes and it revolves around this for 1.75 turns, and nucleosome contains about 200 base pair. That is, since DNA strands are over here, what will happen is that DNA strands will also contain base pairs. So approximately there are 200 base pairs in a nucleosome. So this linker DNA that we see here, that it forms from it moves from one nucleosome to other nucleosome. So that DNA is the region of DNA between two successive nucleosomes, and that is called as linker DNA. And nucleosome along with linker DNA is called chromatosome, not chromosome. That is, its name is very similar to chromosome. It's called chromatosome. We have to study about a few more points also. So since H1 histone was attaching the DNA to the nucleosome, it is also called as linker DNA or a plugging protein. So plugging protein is fixing the incoming and outgoing DNA strands over the body of the nucleosome, and it is the nucleosomes which gives the normal beads on a string appearance. That is, so the normal DNA that we see appears like this. So those red color structures are nucleosomes, and the green color part is the DNA part. So these give the beads on a string appearance. Now we have to study about more compaction. So if that nucleosome part is also further condensed, that is, this part is again coiled upon each other. So what happens is that structure is called solenoid structure, and solenoid structure contains 6 nucleosomes per turn. In physics you must have studied this, something called solenoid, around which wires are located. So it is similar to that. So this is the solenoid structure, and in each turn 6 nucleosomes are present. So we will study the further compaction. So the compaction starts from DNA. So DNA is approximately 2 nanometer. Then we have nucleosome. Nucleosome is 10 nanometer in thickness. Then comes the solenoid structure, and solenoid structure is 30 nanometer. Then comes the chromatin fiber. Chromatin fiber is 300 nanometer in thickness. After it, the chromatid part comes, and then the chromosomes. So its thickness is around 700 nanometers, and the thickness over here is 1400 nanometers. So as you can see, as the DNA is undergoing further and further condensation, we have the thickness increasing from 2 nanometer to the final thickness is 1400 nanometer. So this is the normal sequence of the condensation of DNA. Because of this part, there are two structures that are formed. One is called at further levels, at higher levels of condensation, so histone proteins cannot cause higher level of condensation. So there are some other proteins which are called non-histone chromosomal proteins. So these are the proteins which are causing these higher level of condensation. So because of this condensation, two types of regions in the chromosome appear, and one is called euchromatin and the other is called heterochromatin. So because of the variation in the condensation level, two regions are present. One is called euchromatin. It is loosely packed. It stains light when certain chemicals are induced to it. Then the staining, that is the coloration, is lighter in the case of euchromatin, and it is transcriptionally active. Transcription is a process by which DNA forms RNA. So this euchromatin forms certain RNAs and it replicates earlier. We'll later see that what is replication. Replication is just copying of DNA. So it is replicating much earlier. And the other is heterochromatin. It is densely packed. It stains darker. It is transcriptionally inactive. It does not undergo transcription. It is transcriptionally inactive and it replicates later. So when DNA will undergo a replication, the first structure that will be replicated is the euchromatin area, and thereafter the heterochromatin area will be replicated. So this is the level, this is the main portion of the condensation of DNA. So this is how DNA is packed. So we had already seen that the typical eukaryotic or human nucleus was of 10 to the power minus 5 meter, and here the DNA is 1400 nanometer. This is why, because of the condensation that is occurring, 2.2 nanometers DNA are condensed in the chromosomes of thickness of 1400 nanometer. That is why DNA, even after being such a long polymer, is easily packed inside the nucleus. And this was the whole discussion about packaging of DNA. In the next topic, we are going to start DNA semi-conservative replication.